three.
Hello, everyone, and welcome to today's DDN webinar. I'm Sunita, Assistant Science Editor for DDN, and I will be moderating our discussion. We have an exciting webinar planned for you today. Our speakers, Amar Natarajan and Guangrong Zheng, will discuss the development and applications of PROTACs for cancer therapy. After the talk, Dr. Natarajan and Dr. Zheng will participate in a live Q&A session. To submit your questions or comments, simply submit your questions to the Q&A portal to the right of your screen. We will try to get to as many of these as possible. I'd like to take this opportunity to thank our webinar sponsor, BPS Bioscience. BPS Bioscience's purpose is to advance new scientific discoveries that lead to therapies by creating innovative solutions for research. Our focus areas are immunotherapy, epigenetics, coronavirus, CRISPR, cell signaling, and others, providing 4,000 plus high-quality recombinant cell lines, lentiviruses, proteins, and all aspects of biochemical and cell-based assay development. As a primary manufacturer, we can supply products in large-scale bulk quantities, as well as contract and custom manufacturing. Our biochemical and cell-based screening services are also available to accelerate research. Our sponsor has provided us with some helpful handouts related to today's webinar. You can find and access these in our handouts section, located on the right side of your screen, where you can also find a certificate of attendance for participating in today's live event. And with that, let me introduce you to our first speaker, Amar Natarajan. Amar Natarajan is a Ruth Burnham Professor of Cancer Research at the University of Nebraska Medical Center at Omaha. Natarajan earned an MS in chemistry from the Indian Institute of Technology, Madras, and a PhD in organic chemistry from the University of Vermont. Following postdoctoral training at Harvard Medical School, Natarajan started his independent career in the chemical biology program at the University of Texas. His research is on the discovery and development of target and mechanism-specific small molecule inhibitors/perturbants against disease-relevant targets. Let us take a moment to make sure that the slides are working fine. And with that, Dr. Natarajan, please take it away. Thank you, Dr. Natarajan.
Thank you, Dr. Chari. I would like to thank you and the organizers for this opportunity to participate in this session. So today I'm going to present some unpublished work focused on PROTAC development that targets MET receptor mutants. Let me begin with a brief overview of the two model systems used to target proteins. Inhibitors that reversibly bind to proteins and their efficacy corresponds to the amount of time the inhibitor occupies the protein binding site. Now, these are your classical inhibitors, such as kinase inhibitors. Now, because this is occupancy based, the inhibitor based approach to therapeutics has some inherent limitations. Namely, there is a need to maintain high systemic exposure to ensure adequate in vivo inhibition. Now, this high in vivo concentrations bring with it potential for off-target side effects. And there is a need to bind to the active site, thus limiting the drug target space. PROTACs, which is an emerging paradigm, are a viable alternative that addresses these problems. So PROTACs are heterobifunctional molecules in which a protein of interest inhibitor is conjugated to an E3 ligase binder through a linker. In cells, PROTACs sandwich the protein of interest and E3 ligase to form a ternary complex. Now, this allows the E3 ligase to ubiquitinate the protein of interest. The ubiquitinated protein is degraded by the proteasome in the cell, releasing the PROTAC and E3 ligase. Now, this sets up a catalytic cycle, allowing the next iteration of binding, ubiquitination, and proteasomal degradation. Now, the efficacy in this model system corresponds to the number of times the ternary complex is formed by the E3 ligase, the PROTAC, and the protein of interest. Therefore, this negates the need for high concentrations of drug to in vivo inactivate the target protein. Moreover, since the protein of interest is degraded by the PROTAC, the protein of interest ligand in the PROTAC does not have to bind to the active site, thus expanding the drug target space. The cartoon on the right summarizes the 20-year developmental arc of PROTACs. The first PROTAC was reported by the Cruz and Deschaes laboratories back in 2001. In there, they described the development of a methionine aminopeptidase 2 targeted PROTAC or MET-AP2 PROTAC. They used ovalocin as a MET-AP2 binder and a peptide derived from the protein I kappa B alpha, which, when phosphorylated, recruits an E3 ligase beta TRCP to ubiquitinate the target protein. A major inflection point in this arc from a drug discovery standpoint for the PROTAC field was the discovery of cereblon as the target of IMiD drugs, namely thalidomide, pomalidomide, and lenalidomide. Alongside, the Cruz lab developed a small molecule that binds to the E3 ligase VHL. Now, proof of concept studies that used the IMiDs, namely thalidomide, pomalidomide, or lenalidomide, and the VHL ligand to generate viable PROTACs was demonstrated in 2015 independently by the Cruz lab and the Bradner lab. This enabled the chemistry community to begin conjugating an array of protein binding molecules to the well characterized E3 ligase ligands to develop PROTACs against a variety of targets. Now, a major milestone or proof of concept from a therapeutic perspective is the entry of PROTACs into the clinics. As of now, there are 12 PROTACs that are in clinical trials, with ARV-110 developed by Arvinas that targets the androgen receptor, and ARV-471, again developed by Arvinas, that targets the estrogen receptor, have advanced to phase 2 clinical trials for prostate and breast cancer, respectively. Here, I would like to highlight DT2216 that selectively induces the degradation of B-cell lymphoma extra large, or Bcl-XL, which was developed by Dr. Zheng, the next speaker. Incidentally, DT2216 happens to be the only PROTAC that is currently in clinical evaluation which uses a VHL binding ligand. Now, with that brief introduction, I would like to focus the rest of my talk on the protein MET. So MET is also known as hepatocyte growth factor receptor. MET has been a challenging target in oncology despite its known oncogenicity. MET mutations or MET amplifications can be de novo oncogenic events or can be acquired at the time of resistance to other therapies. MET amplification and MET mutation accounts for about 10 to 15% of non small cell lung cancers. Now, mutations in the donor or acceptor splice sites of MET exon 14 can lead to skipping of exon 14 during splicing. Now, this results in the fusion of exon 13 with exon 15. Now, translation of the spliced mRNA results in a truncated MET that lacks the juxtamembrane domain. The juxtamembrane domain in MET has a degron recognized by the E3 ligase Sybil. Therefore, the loss of the juxtamembrane domain leads to increased stability of the truncated MET or the mutant MET. Now, to assess the change in the half-life of mutant MET, the Cruz lab subjected triple negative breast cancer cells, MDA MB231, that contains a wild type MET, and a gastric cancer cell line, HS746T, that contains a MET exon 14 skipping mutation, to cyclohexamide chase experiments. They demonstrated that both under basal and HGF activated states, exon 14 skipping significantly increases the stability of mutant MET. Furitinib, shown in blue, is a non-selective kinase inhibitor that binds to over 130 kinases. The Cruz lab developed a furitinib-based PROTAC SJF8240 and characterized its efficacy for targeting MET. They used the VHL ligand, shown in red, to target the E3 ligase. The furitinib-based PROTAC SJF8240 binds to approximately 50 kinases, but degrades less than 15 of those bound kinases, which includes MET. Now, this is consistent with previous reports by us and others that showed converting an inhibitor to a PROTAC improves selectivity. So recently, FDA approved tyrosine kinase inhibitors, capmatinib and tepotinib, as first-line treatments for non-small cell lung cancer patients harboring MET exon 14 skipping mutations. Now, since MET exon 14 skipping mutation results in the loss of the degron, namely the juxtamembrane domain that is required for recognition by Sybil and subsequent ubiquitination, we hypothesized a MET targeting PROTAC using a selective MET inhibitor would restore ubiquitination of MET with exon 14 skipping mutations and promote its degradation. To test this hypothesis, we generated a library of PROTACs with an array of head groups targeting different kinases. It includes MET inhibitors and E3 ligase ligands based on the IMiDs and the VHL binding ligands. Now, the two were conjugated using an array of linkers such as PEG, mixed PEG, carbon that have different lengths and different compositions. Now, to screen this library, we co-opted a strategy reported by the Gray, Fisher, and Westover laboratories about a year ago. So they used cells expressing a GFP fusion protein to evaluate degraders by using the loss of GFP signal as a surrogate for target protein degradation. Specifically, they were interested in developing KRAS G12C targeted degraders. They used a GFP KRAS G12C fusion protein expressing reporter cells to screen the PROTAC library. The PROTAC library consisted of quinazoline scaffold built on ARS 1620, a covalent inhibitor of KRAS G12C. Now, the screen identified a KRAS G12C targeted degrader XY488. XY488 exhibited a loss of GFP signal in both a dose response and a time course study, with maximal degradation at 1 micromolar of XY488 at 24 hours. Although this strategy failed to successfully develop a PROTAC that degraded the native or endogenous KRAS G12C, the screening strategy itself appealed to us. So we generated a pair of cell lines that stably expressed either a GFP vector or a MET exon 14 skipping GFP fusion protein. We opted to conduct a live cell imaging screen with our PROTAC library at 1 micromolar. So this is a representative example of wells treated with either the vehicle control or the PROTAC is shown. Now, this is a time course study where the cells were imaged every 2 hours for the loss of GFP signal. And as you can see, the PROTAC signal, PROTAC treated cells, the GFP signal is almost completely gone after 24 hours. And we extrapolated the loss of GFP signal to the destruction of the fused mutant MET. So we quantified the change in GFP signal over time and calculated the percent degradation of MET exon 14 skipping mutant over time. Curve fitting the data allowed us to determine what we termed as DT50. So DT50 corresponds to the time it takes to observe a 50% loss of GFP signal in cells treated with PROTAC. The assay exhibited a rather large dynamic range, allowing us to rank the PROTACs based on their DT50 values. For example, the study showed 48-279 as an inactive PROTAC, while 48-278 was partially active, and it allowed us to identify a potent degrader in 48-284 that had a DT50 of about 5 hours. We also conducted a dose response study under the same paradigm where we screened compounds at different concentrations ranging from 0 to 10 micromolar. And we were able to show that PROTAC 48-284 was able to degrade GFP effectively at about 0.9 micromolar, and the degradation dropped off on at either lower or higher concentration. Now, this is a classical feature of PROTAC and is called hook effect, wherein increasing concentrations of the PROTAC leads to the formation of stable binary complex between the PROTAC and the target molecule, or the PROTAC and E3 ligase. This prevents the formation of the ternary complex, which is required for its efficient degradation. That is why we don't see efficient degradation at high concentration of the PROTAC in this particular graph. Now, to assess if the degradation of MET exon 14 skipping mutant induced by 48-284 is mediated by the proteasome, we subjected the fusion expressing cells to 48-284 in the presence and absence of MG132. The loss of GFP signal induced by 48-284 was blocked when MG132 was co-administered, demonstrating that the degradation is proteasome dependent. To confirm degradation of metexon 14 skipping GFP fusion protein and assess the proteome wide selectivity of the PROTAC 48 284, we subjected the GFP expressing cells to 1 micromolar of either the MET inhibitor or the PROTAC 48 284 for 24 hours. And the lysates were subjected to mass spectrometry based proteomic analysis. The plot here is a principal component analysis of the vehicle inhibitor and PROTAC samples, and as you can see, they cluster separately. The volcano plot on the right shows that approximately 5,500 proteins from these lysates were quantified, and 45 of the 5,501 protein exhibited greater than twofold reduction in the abundance ratio with a p-value less than 0.01. Now, these were classified as hits. Among the 5,500 proteins quantified, there were 119 kinases that were also quantified. And of these 119 kinases, only 4 kinases, including MET, had an abundance ratio of less than 2 and had a p-value less than 0.01. Now, the 4 kinases are shown here in red dots. The parental MET was also degraded in addition to the fusion protein. The 3 other kinases that were degraded by 48 284 were PDPK1, EPHA2, and PIK3R4. Now, a recent study showed that PDPK1 is associated with MET, which suggests that PDPK1 degradation could be an indirect effect. Next, we did a head-to-head comparison of PROTACs derived from a MET inhibitor or a non-selective inhibitor, which is SJF8240. In the dose-response head-to-head comparison study, we observed that 48 284 was able to degrade the MET exon 14 skipping GFP at lower concentrations when compared to the non-selective furitinib-based PROTAC SJF8240. Once again, we observed the hook effect where the most efficient degradation happened at 1 micromolar and the efficiency dropped at lower or higher concentrations of the PROTAC. These studies also show that 48 284 derived from a selective MET inhibitor was more than twice as efficient at degrading the mutant MET when at half the concentration of SJF8240. Next, we examined the effect of 48 284 using HS746 T cells that expresses native or endogenous MET exon 14 skipping mutation. In the time course study, treatment of HS746 T cells with 1 micromolar of 48 284 showed a time dependent decrease in the mutant MET. In a dose response study, we observed a dose dependent decrease in the levels of mutant MET protein. Importantly, we observed the hook effect above 1 micromolar. Once again, 48 284 more efficiently degraded mutant MET when compared to SJF8240 in both the time course and the dose response study. Previous studies have shown that mutant MET increases and prolongs RAS-AKT and RAS-ERK signaling. To determine if 48 284 and SJF8240 affect these pathways, we looked at phospho-AKT and phospho-MAPK levels in these treated cells. Our data shows significant reduction in both the phospho-AKT levels and phospho-MAPK levels in 48-284 cells. These data show that 48-284 is a potent degrader of mutant MET and disrupts downstream signaling in a cell line with native or endogenous MET exon 14 skipping mutation. Next, we assessed the degradation of the target, which is MET exon 14 skipping mutation by 48-284 in a short term UW21 xenograft model. UW21 xenografts were derived from brain metastasis of a non small cell lung cancer patient, and it contains a MET exon 14 skipping mutation. Briefly, UW21 was implanted into the flanks of mice and the tumors allowed to grow for 2 weeks. Half the mice were treated with 48-284 through tail vein injection twice, 8 hours apart, and the tumors were removed 6 hours after the second injection. Now, when compared to the untreated controls, immunohistochemistry studies showed that the treated tumors showed significant reduction in MET expression. Lysates from the UW21 treated xenografts also showed higher molecular weight species of MET, which were absent in the controls, suggesting 48-284 mediated ubiquitination of the mutant MET and potential degradation. We also tested 48-284 in a PT425 3D cancer spheroid model. Now, this model has an oncogenic PTPRZ1 MET rearrangement, also known as ZM fusion, that occurs in gliomas. Now, PT425 was derived from a resected glioma, and sequencing identified exon 2 of PTPRZ1 joins exon 2 of MET, which is in the 5' UTR region of MET. Now, this fusion is predicted to result in an in-frame fusion of 5 novel amino acids between exon 2 of PTPRZ1 and the start of the coding region of MET. The PTPRZ1 promoter drives the transcription of MET in these fusion rearrangements, which retains the MET dimerization and phosphorylation properties. So, in the PT425 3D spheroid model, 48-284 had an IC50 of 100 nanomolar. We are currently conducting ADMET and PK studies to continue advancing 48-284 through the drug discovery process. Let me quickly summarize what I showed you today. So, our studies show that MET targeting PROTACs can be rapidly identified by live cell high throughput imaging using a MET exon 14 skipping GFP expressing cell lines. Now, these PROTACs can restore ubiquitination of MET that is lost with exon 14 skipping in native models that express endogenous MET mutants. And this may represent a therapeutic strategy not just for one type of MET mutant, but multiple types of MET alterations. With that, I have a long list of people to thank. So, this was a collaborative project between my lab in Nebraska and the Mayo team led by Dr. Aaron Mansfield. So, the study was spearheaded by Dr. Mallareddy in my lab and Dr. Lin in Dr. Mansfield's lab. And we are grateful to all the collaborators at both institutions that contributed to the project. I would specifically like to acknowledge the Halleck Family, Met Crusaders, Mayo Clinic's SIM and PCT programs that supported this work. This work was also supported by a University of Nebraska collaborative initiative grant, an NIH grant that supported part of the work and the course along with training of graduate students. With that, I would like to once again thank you for the opportunity, and I look forward to hearing Dr. Zheng's talk.
Thank you, Dr. Natarajan. Thank you for a very illuminating talk, for the introduction to the PROTACs field, and thank you for sharing the preclinical studies coming out from your lab targeting MET mutants using a very specific MET PROTAC inhibitor. And now on to our second speaker. Guangrong Zheng is an associate professor in the Department of Medicinal Chemistry at the University of Florida College of Pharmacy. His group focuses on the discovery and development of targeted protein degraders for cancer treatment. Zheng received his PhD in organic chemistry from Shanghai Institute of Materia Medica and his BS in medicinal chemistry from Fudan University in Shanghai, China. Let us just give a second to make sure that the slides are working fine. Dr. Zheng?
Let me try to share my screen. Can you see my screen?
Yes, so we can see your screen. Thank you, Dr. Zheng. Take it away.
Okay, thank you, Sunita, for the introduction and also for the invitation. I really enjoyed Dr. Natarajan's talk. So he actually set up the stage for my talk, so I don't have to repeat out the PROTAC concept. So I'm going to briefly talk about our Bcl-XL, Bcl-2 PROTAC project, and also show you how we use the PROTAC technology to achieve the reduced on-target toxicity of Bcl-XL inhibitors. So this is my disclosure slide. So this project is in collaboration with Dr. Hong Zhou's lab since 2015. And a number of other labs around the nation. I would like to thank all the team members and the collaborators who have been working on this project for these years. And this is truly a great team effort. So as Dr. Natarajan just mentioned, PROTAC is really a paradigm shifting drug discovery technology. He already nicely introduced the concept of PROTAC and its potential advantages over small molecule inhibitors. So I'm not going to repeat. In reference to the project I'm going to talk about, so I want to emphasize that PROTAC approach could be used to increase tissue selectivity based on the differential E3 expression in different tissues or cell types. So the hypothesis of using PROTAC to increase tissue or cell type selectivity is quite straightforward. That is, the E3 ligase expression level in cell should correlate with the protein degradation efficiency of the PROTAC that recruits this E3. So high amount of E3 ligase in the cell should translate into faster, more potent, and deeper degradation of the protein, and vice versa. So to further elaborate, if you have a small molecule drug able to do its job by inhibiting a specific disease-causing protein in the disease tissue, if this specific protein also plays an important function in a normal tissue, so this drug will cause on-target toxicity to the normal tissue. So if you can find an E3 ligase that is lowly expressed in the normal tissue and highly expressed in the disease tissue, then if you can find a ligand for this E3, then you potentially can build a PROTAC based on the small molecule drug. Then because the lower expression will translate lower toxicity, high expression of E3 may translate to higher potency. So you will be able to increase the therapeutic index. Okay, so by analyzing the publicly available data sets, so indeed the tissue and the cell types enriched or specific E3 ligases are available. So this figure is from a recent review by Cruz Lab. It shows some examples of tissue-enriched E3s and some tumor-enriched E3s. So this topic has been extensively covered by a number of reviews. So, so this E3s actually give us huge opportunities for developing PROTACs that can be specifically or more selectively targeting disease. So we can call those PROTAC as a precision PROTAC. However, it could be very challenging to develop a E3 ligase ligand that is based on an unknown E3 ligase ligand, E3 ligase. So in order to have the ligand be able to convert to be used for PROTAC development, we need to have a very good understanding of the mechanism of action of these E3. Also, the E3 may not have the structural information. So also the E3 may not have a good binding pocket. Also, maybe developing the assay for drug screening is not very easy. So in our case of targeting Bcl-XL, we may have a unique opportunity actually to utilize the existing E3 ligase ligand, use this technology to reduce the on-target toxicity of Bcl-XL inhibitors. So Bcl-XL belongs to the Bcl-2 family of proteins that are key regulators of the intrinsic apoptotic pathway. In other words, they decide cell death. So this protein family includes anti-apoptotic proteins and pro-apoptotic proteins. Anti-apoptotic proteins such as Bcl-2, Bcl-XL, and Bcl-1 all have BH1, BH2, BH4 homologic domains. Pro-apoptotic proteins have two varieties, including the multi-domain backs and back, and a larger number of protein called the BH3 only protein. They have only one domain called BH3 domain. So this is a diagram showing how Bcl-2 family proteins work. Back's back can form oligomers on the mitochondrial membrane and act like punching holes on the mitochondria, which cause the release of cytochrome C, then trigger the apoptosis cascade. So anti-apoptotic protein binds to backs and back to prevent them from forming oligomers. BH3 only protein can bind to anti-apoptotic Bcl-2 proteins to release backs and back. Some of them can also directly activate backs and back. The interaction among these three groups of proteins determine the fate of the cell. So cancer cells are often under apoptotic stress, so they often depend on anti-apoptotic Bcl-2 proteins to resist apoptosis. So anti-apoptotic Bcl-2 proteins are important anti-cancer targets. Among these proteins, Bcl-XL is the most commonly overexpressed anti-apoptotic Bcl-2 proteins in cancer cells. Also, they appear to be mainly responsible for chemotherapy resistance. So you inhibit Bcl-XL, you could sensitize chemotherapy. So my collaborator, Dahong Zhou's lab, also a number of years ago, they also identified Bcl-XL is a key survival gene for a number of senescent cells. So targeting senescent cell is an approach to treat a number of aging-related diseases. So many BH3 mimetics that basically mimic the BH3 only protein function. So those BH3 mimetics have been developed as inhibitors of Bcl-XL, Bcl-2, and MCL1. So Bcl-2 selective inhibitors, venetoclax, also known as ABT-199, has been approved by the FDA for the treatment of a number of hematological malignancies, including CLL and AML, which really validate the approach of targeting anti-apoptotic proteins. So shown here are some of the Bcl-XL, Bcl-2 dual inhibitors, and some well-known Bcl-XL selective inhibitors. But unfortunately, it was found that platelets heavily depend on Bcl-XL for survival. For example, as shown in this cancer research paper, so ABT263, also known as navitoclax, this compound has been phase 2 clinical trials. So ABT263 causes rapid and dose-dependent decrease of platelet counts in dogs. So we used MTS cytotoxicity cell viability assay. We compared a series of Bcl-XL inhibitors, including some of our own analogs, for the selectivity between human platelets and multiple cells. So multiple is a T cell acute lymphoblastic leukemia cell line. These cells depend on Bcl-XL for survival. So you can see none of this compound actually have any selectivity. So traditional structure optimization approach won't design out this on-target toxicity. So we think the PROTAC is a potential approach to tackle this problem. It is not so surprising that protein levels of E3 ligase are low in platelets. Showing here are PHL and the CRBN. Both are commonly used for E3 ligase for PROTAC design. In addition, E1 and the E2 ubiquitin activating enzyme, E2 is ubiquitin conjugating protein. They're all low in platelets. So as I mentioned earlier, low levels of E3 ligases could make the corresponding PROTACs less effective. So converting Bcl-XL inhibitor to Bcl-XL PROTAC degraders, we could see reduced on-target platelet toxicity. So we started an initial study. So after that, we actually found the proof of concept. We really see protein degradation of Bcl-XL and also reduced toxicity to platelets. So we then start a pretty extensive medicinal chemistry campaign by screening different combinations of Bcl-XL ligand, we also call warheads, linkers, and E3 ligase ligases and ligands. We use Bcl-XL degradation, mostly Western blot. We also have the fusion protein use GFP, just as Dr. Natarajan just mentioned, similar method. So we also use the selectivity cytotoxicity assay, just compare the toxicity between platelets and the multiple. So this as a primary assay. So eventually we select this compound named as DT2216 as a development candidate. This compound is now in phase 1 clinical trials. So I just want to talk some data around this compound. So DT2216 acts as a Bcl-XL degrader, dose dependently induce Bcl-XL degradation in multiple cells. However, there's no obvious degradation in platelets after 16 hours treatment, indicating the success of this design concept. Compared to its warhead, DT-AB2263, DT2216 is more potent in inhibiting multiple growth. As I mentioned earlier, multiple cell depend on Bcl-XL for survival. So importantly, DD2016 is not toxic to platelets up to 10 micromolar. So this degradation is time dependent. Uh, it's pretty fast, but not we have a compound actually can degrade within 30 minutes. So this is not very fast, but it's very amazingly fast. Within 8 hours, you will see almost complete degradation. The degradation seems also lasts long time. So after treat the cell for 16 hours, you wash away the compound, then up to 48 hours, you still see Bcl-XL reduction. So here are some of the typical experiments performed to basically confirm the mechanism of action of this compound is indeed a PROTAC. So the Bcl-XL degradation is depend on the E3 ligase. As you can see, when you co-incubate with BHL ligand, you can block the protein degradation. Also, if you treat cell without BHL or BHL non-exist or knockout, you don't see degradation. So we also synthesize negative control in which the BHL ligand used diastereomer replaced by a stereomer. So this compound does not bind to BHL. You can see negative control does not induce Bcl-XL degradation. As a result, the negative control has much lower cytotoxicity to MOD4, indicating the cytotoxicity of DD2016 is mainly derived from Bcl-XL degradation. So the degradation also depends on proteasome. As you can see, proteasome inhibitor MG132 can rescue the degradation. Also, the warhead ABT263 co-incubated with this warhead, you can rescue the degradation, indicating the degradation require occupate engage with the target. Okay, so we move into in vivo studies. DD2016 has low clearance rate, and the plasma elimination half-life is about 6 to 10 hours. However, it has sustained low ability lowering of Bcl-XL. You can see the somehow the compound can accumulate in tumor tissue for about a week after IP injection of 15 milligram per kg of DD2016. And the Bcl-XL degradation was observed over 2 weeks after 0.1 IP injection of 15 milligram per kg. So moving to efficacy studies, we have done multiple cell line derived and patient-derived xenograft models. Showing here are studies multiple xenograft model. As you can see, 50 milligram per kg once a week IP injection can almost completely inhibit tumor growth, which is more effective than once daily 50 milligram per kg oral dosing of ABT263. DD2016 also less toxic to platelets in vivo compared with ABT263. So in the tumor tissue, we also observed Bcl-XL degradation, but we did not see Bcl2 degradation. Okay, so in addition to what I just presented, that degradation of Bcl-XL by PROTACs can directly kill tumor cells. Our collaborator, Dr. Zheng's lab, also found that tumor-infiltrating regulatory T cell are also primed to Bcl-XL inhibition or degradation for the induction of apoptosis. So I don't have time to present that data, but DT-2016 induces apoptosis of tumor-infiltrating Tregs and activate tumor-infiltrating CD8 positive T cells. So those actually can enhance anti-tumor immunity. We see in four different syngeneic models. As I mentioned earlier, Bcl-XL also a key survival factor of senescent cells. So accumulation of senescent cell has been shown to play an important role in tumor initiation, relapse, and the metastasis. So the ability of this compound DT-2016 in clearing senescent cell could benefit cancer treatment. So all these findings suggest that DT-2016 has the potential to be developed as a novel anti-cancer drug. So DT-2016 is derived from ABT-263, which is a dual inhibitor of Bcl-XL and Bcl-2. So you can see DT-2016 combined to Bcl-XL, Bcl-2. However, based on the Western blot assay we use for many cell lines, and also the global proteomics assay, we never see Bcl-2 degradation, and so this compound is a specific Bcl-XL degrader. So as a result, DT-2016 is not very effective as a single agent on cancer cells that depend on Bcl-2 or both Bcl-XL and Bcl-2. For example, in this PDX model, T-cell lymphoma PDX model, because the cancer depend on both Bcl-XL and Bcl-2. So the DT-2016 treatment is showing in the red dot. It's not very effective as single agent, but when it's combined with the Bcl-2 selective inhibitor, ABT-199, you can see the purple dot. They're very effective. So if we can develop a dual degrader, that'll be improve the anti-cancer spectrum. So that's why we try to understand why this compound does not degrade Bcl-2. When you compare the structure of Bcl-XL and the Bcl-2, you can see the Bcl-XL has more lysine residue available for ubiquitination. So through a series of mutagenesis assay and the mass spectrum analysis, we found that the K87 lysine unit is being ubiquitinated on Bcl-XL, but in the same region, there's no lysine available. So we think maybe lack of lysine here may be the reason. In addition, we also used the nanobread assay in cell. It was found that Bcl-2 does not form ternary complex with this compound does not induce Bcl-2 and the VHL ternary complex. So we don't know the reason, but seems this also contributing to the non-degradation of Bcl-2. Because form ternary complex also is required for protein degradation. So it seems like if we can maybe select different orientation, use a different exit vector to target this protein, maybe we're able to reach lysine, for example, K17, and also create a new protein-protein interaction surface that allows us to form a stable ternary complex. So we explored the different exit vectors on both ABT-263 and VHL ligand. So obviously DT2016 is linked from here. So we found actually if you link from this position, we're able to develop compound can degrade both Bcl-XL and Bcl-2. So this compound PZ18753B we recently published can degrade both Bcl-XL and Bcl-2. So as shown here, the two compound PZ18753B is able to degrade both Bcl-2 and Bcl-XL, and compared to DT2016, you don't see degradation of Bcl-2, but you do see degradation of Bcl-XL. So as a result, PZ18753B can more potently kill multiple cell, which is dependent on Bcl-XL. Also, it can more potently kill Bcl-2 dependent IS4 cell. And in this cell line, H146, small cell lung cancer cell line, you see this depend on both Bcl-XL and Bcl-2. The compound also works. So we also conduct preliminary in vivo assay on this compound. So in multiple xenograft model, 753B is equally effective to DT2016, but with about 6 times lower dose. So in the H146 small cell lung cancer model, 753B is effective as a single agent. We previously did on the same model with DT2016. So single agent is not really effective. So you will have to combine DT2016 and ABT199, the Bcl-2 selective inhibitor, to have this similar effect. Okay, so I want to quickly summarize my talk. So Bcl-XL is an important high-value cancer target because the proteotoxicity is on-target toxicity. Traditional inhibitors cause the thrombocytopenia, which prevent their clinical use. So we demonstrate here the use of PROTAC approach is effective in reducing on-target predatory toxicity of Bcl-XL inhibitors. So we think this approach could be applied in other cases, in case you have an on-target toxicity. So if you can find differentially expressed E3 ligases and the ligand, if the ligand can be found for that E3. So DT2016 is a specific Bcl-XL degrader that can effectively inhibit tumor growth in vivo, but without causing significant thrombocytopenia. DT2016 can also inhibit Bcl-XL independent cancer by selectively killing tumor-infiltrating regulatory T cells. So we think DT2016 may be developed as a first-in-class anti-cancer PROTAC targeting Bcl-XL. Also show some data on Bcl-XL, Bcl-2 dual degraders. We think could have a much broader application in cancer treatment. So we are now in the stage of selecting a IND candidate for further development. All right, thank you all for your attention, and I will be happy to answer any question you may have.
Thank you, Dr. Zheng, for sharing with us your research on Bcl-XL and how it is an effective PROTAC inhibitor. In the time that we have now left, we can get to our listeners' questions. I will start off with more of I think, a general question to both of you, but I'll let probably Dr. Natarajan have a stab at this because he did talk about the introduction to the field. So to me, as a person who's just getting into this understanding the PROTACs thing, it looks like there's a lot of design that has to go in before you can target your PROTACs. For example, you have ligand specificity, and then you have E3 ligase specificity, then you have protein of interest specificity, and you have to actually combine all of these things together before you're able to come up with a PROTAC which is efficient for the particular protein of interest. So, Dr. Natarajan, could you just walk us through some of your initial thoughts when you go about designing your PROTAC?
Yeah, you hit on all the key variables. So you need a ligand that binds to your protein. It seems that based on the analysis of a whole bunch of PROTACs that have been synthesized, uh, the ligand selectivity is not as important, or the E3 and the E3 ligase ligand selectivity is more important, and the linker length and linker composition seem to play a bigger role in the efficacy of a given PROTAC. A design perspective, as a chemist, I'm always looking for a protein that has a known ligand.
Could you just repeat that, Dr. Natarajan?
Yeah, so you need, if you want to design a PROTAC, the key is to find your ligand that binds to your protein of interest.
Mhm.
And once you have that, right now we cannot use first principles to say this linker length and this linker composition is going to work. Beyond that, it is essentially trial and error. You build a library and you identify a PROTAC that works, and then you go out and optimize that using a medchem strategy. And our experience is it takes about 30 to 35 PROTACs to find one that actually works, that needs to be optimized subsequently.
Great. Thank you, Dr. Natarajan. And following off from that, Dr. Zheng, you did touch upon this in your talk. You talked about selecting the E3 ligase, for example, choosing E3 ligase, which is possibly expressed more in a diseased tissue versus the healthy tissue. So, when you went about designing your Bcl-XL, could you tell us a little bit about how you went about the strategy and what are the E3 ligases that you looked at, and how we finally zeroed in on the E3 ligase to target?
Sure. Yeah. So obviously, as I mentioned, the problem of this target is specifically on-target toxicity to platelets. So in this case, it's a bit easier, right? You just need to avoid platelet toxicity. So in normal design, you will have to select E3 ligase actually highly expressed in the cancer. For example, whether we targeting the cancer type selective overexpress E3. But in our case, obviously the platelets is not a whole cell, so they don't have a nucleus, so they don't have the requirement for like protein recycling. Um, so that's why they have very weak ubiquitin-proteasome system. So we can take advantage of that to use the supposedly ubiquitous expressed E3, such as CRBN and VHL, to design PROTAC for this target. But other target, you will have to select E3, which is restrict in certain tissues or certain cell types.
Okay. Thank you for that. And also, Dr. Natarajan, when you were talking about the PROTAC and you were showing us the enzyme kinetics of how quickly the PROTAC is able to degrade the protein of interest, you did mention the timelines ranging from like 22 hours to 5 hours. And I was wondering, a PROTAC what is the half-life of it, and how stable is it once it is inside the cell?
That's a good question. We have not tested our PROTACs for stability in cells, um, but with all small molecules, it probably has the same stability issues. If you have metabolic liability in your molecule, you're going to have stability issues, which will usually get cleaned up in the second round of synthesis and screening.
Great. Uh, Wonderful. Thank you so much. We do have some time for some audience-related questions. So one of the questions that's coming into us is, do you think that this technique can be used to target viral and microbial proteins to design antivirals and antibiotics? I think I'll let one of you decide who is going to answer that, but this is a question. It's a more of a general question. If this can be used for targeting antivirals and antibiotics.
It comes down to E3 ligase and ubiquitin-proteasome system in the microbe or the virus. So if you don't have that, then you cannot get rid of the virus or the microbe.
But I mean, most of the virus and bacteria do have. I mean, viruses especially hijack our genomic system. So, isn't it possible to possibly then design a PROTAC more easily for antivirals as opposed to bacterial?
Yeah.
True. You can get rid of if you take COVID as an example, MPRO is the enzyme that is used to cleave the viral polypeptide. So if one were to use an MPRO inhibitor and design a PROTAC against that, that could be a viable strategy as a COVID therapeutic.
Dr. Zheng, would you like to add something to that?
Yeah. So as Natarajan mentioned, yeah, COVID as a case, a lot of people actually developing pro degrader because you can hijack the E3 ligase in the host cells, right, to degrade the virus protein. And also it's been demonstrated in, I believe it's HBV or hepatitis virus. It works. But for the bacteria, because they have their own system, I don't think they have the same ubiquitin-proteasome system. They may have different protein recycling system that need to be separately developed.
But once you have a bacterial infection, I'm just riffing off of this question, but once you do have a bacterial infection and once it is in the host, can we then not target the protein of, is the protein that is being synthesized by the bacteria, target that to a E3 ligase with the host? Is that possible?
Yeah, I guess theoretically, if the bacteria actually in the host cell, then you will probably able to target on the surface, bacterial surface proteins or something, because we do see aggregates, like protein aggregates or organelles can be degraded by this approach.
Great. Thank you. Another question that we have, I think this is to Dr. Natarajan. When you talked about the in vivo studies and injecting the PROTACs into the mice, do you use a delivery system? And if that is, what is the vehicle that was used to deliver the PROTACs?
No, we have an in-house formulation that keeps these PROTACs in solution, and we use that formulation to IV dose the mice.
Okay. And then, so this is another question that I had. When you talk about, it's again, it's a general question, so I'll probably let both of you take turns. When you deliver the PROTACs, how is it a more of a general delivery, or is there a way to specifically target that to the tumor tissue and tumor cells expressing that particular protein of interest? This actually becomes more pertinent to Dr. Zheng when he was talking about the toxicity, but I think it's more like, a general question as to how do you target the PROTACs to the site where it's needed?
Yeah, that's a good question. I think it's really no difference from traditional small molecule inhibitors in terms of targeting, but it's possible if you have E3 actually higher enriched in a tissue, maybe that will drive the enrichment for that PROTAC to that tissue. Otherwise, it's probably similar to small molecule inhibitors. You can also design, antibody conjugates, or maybe pro-drug approaches that you can allow you to targeting certain tissue or cell surface proteins. Yeah, that but I don't think that you actually, we see a lot of evidence actually you can distinguish PROTAC and the traditional small molecule inhibitors. But we do see actually tissue accumulation of the PROTAC somehow.
You see, sorry, Dr. Zheng, you see tissue accumulation. Yes, that is very true. So can you talk a little bit about, your graph where you showed that the PROTAC is enriched in the tumor over a course of time? Is it like, did you do any stability studies? Did you do any activity studies to see if the protein was getting degraded? What was actually happening in that particular graph? What were the events that were happening in that particular graph that you showed us?
Yeah, we did both. Basically, we take the tumor, we divide in half, then measure the drug concentration, then measure the protein level. So we see there's accumulation of the drug in the tumor tissue. Also, there's a delay, longer duration of protein degradation. I think that's a lot to do with the catalytic nature of the compound, because you actually can still degrade the protein when the drug concentration is very low, because it's catalytic mechanism of action. So we do see accumulation over a week, then you see degradation over 2 weeks in one study. So I also see other study. You do see the PK PD decoupling phenomena. It's kind of similar to the covalent inhibitors.
So when it's accumulating in the tumor, going back to Dr. Natarajan's talk about having an optimal concentration, because if you don't have an optimal concentration, they can either form a binary complex with the protein of interest or with the E3 ligase. How do you actually, and I'm assuming at this point that if it's just accumulating in the tumor tissue, the concentration is going up and up. Do you see any of those binary complexes forming? Do you see any, the activity of the PROTAC being altered because they're raising concentrations?
I think that's a question for Dr. Zheng, right?
Yeah, it is. Yeah.
So this is an interesting question, because you often see the hook effect in cell culture, but I never see anybody report this in vivo. Obviously, maybe it's difficult to really study that, but certainly we don't see that in our case. Yeah, maybe basically the compound won't be, the concentration won't be high enough to have that effect in vivo.
I mean, the other thing that happens in vivo is it comes down to your readout, right? So
Right.
your compound can still function as an inhibitor, not just yours. I mean, even our
Yeah.
compound can still function as an inhibitor. So at higher concentrations, they're probably functioning as inhibitors.
Exactly. Yeah, you still have the same outcome, but you don't know, maybe a mixed mechanism.
Yeah.
True, Dr. Natarajan, they are acting as inhibitors because they're inherently inhibitors, and then they become PROTACs. But I was wondering about the activity being modulated by the formation of binary complexes. So I think the definitive answer is that you can't really see that in vivo because they are not enriched to that level. Am I understanding that right?
I don't know. It's kind of, I don't know. I don't recall seeing a specific paper where they did a dose response in a mouse model, because you need a large enough dose concentration to pick.
Concentration.
Yeah,
Okay.
you can do that in a cell culture environment, but I'm not sure, because you're working with this tox versus efficacy window, right? If you go
Okay.
too high, the compound might be toxic inherently.
Okay, thank you, Dr. Zheng and Dr. Natarajan. Another question that we have, and this is for Dr. Zheng, are there safety observations with the BCXL2, BCXL/2 in addition to that seen in the platelets?
We haven't we haven't looked into the toxicity yet, but based on the Bcl-XL specifically inhibit PROTAC, we don't see any other toxicity besides the platelet toxicity.
Okay. And then, do you have any comments? I think this can go to Ivor. Do you have any comments on the ADMI properties when the molecule is getting bigger?
Yeah, obviously the ADME is terrible, right, for this molecules, especially for our molecule. This molecule weight is above 1500, so it's the water solubility and the permeability supposed to be very poor. However, the catalytic nature of modal action may actually overcome that poor admin property. But for some other target, maybe you actually can design PROTAC, which is very close to traditional small molecule inhibitors at 600 or to 900 molecule weight. So those molecules actually you can do oral dosing.
Okay, thank you. Dr. Natarajan, so you showed us wonderful preclinical data supporting the MET inhibitor PROTAC. What are your future plans for this? Are we looking at clinical trials? What are the next steps for this PROTAC?
Yeah, we are moving in that direction. So we are starting to do some PD studies now that we have proof of concept, and then we'll probably do some IND enabling tox studies. We have a couple different candidates to choose from, but yeah, the goal is to get into clinical trials.
Great, thank you so much. That should be exciting. And Dr. Zheng, how about with the Bcl-XL PROTAC and also the specific inhibitor that inhibits both Bcl-XL and the Bcl-2? What are your future plans for that one?
Yeah, so we the first compound already in clinical trials, so we hope that will move forward, more advanced in clinical.
Is it in phase 1 right now?
Yeah.
Okay.
So for the newer generation, we currently selecting a candidate for IND enabling studies.
Can you tell me a little bit more about that?
Yeah, so basically choose a compound to go for the manufacturing, chemistry, manufacturing control, and the toxicity studies, and a bunch of efficacy studies to have a package for FDA to clear for us to move to human trials.
Okay. Okay, I think that we have gotten around to all of the questions that we had for both Dr. Zheng and Dr. Natarajan. If there are no further,
I see several questions in the chat.
Oh yeah, I'm so sorry. Yeah. Yes. I'm so sorry. Yes. So what about the covalent ligands for the POI? Is there any advantages or disadvantages for using such a ligand for building PROTACs?
It depends on what side of the molecule is covalently bound. So if you have an E3 ligase binding covalent ligand, that's good, because then you make it a binary reaction. But if your covalent ligands are targeted to the POI, as the person asked, that has no specific advantages, because now you're killing off the catalytic nature of the PROTAC.
Yeah, I think I'll have to unpack it a little bit more. So we are talking about, instead of linkers, we're talking about covalent ligands to the POI. Okay, so yeah.
If you do that, when your POI is degraded,
yes,
you're not going to get your PROTAC back, because you're going to end up with a modified PROTAC it's not going to be able to bind to another protein of interest. So you're killing off the catalytic nature.
Yes, because you need the PROTAC to be as a catalyst. It's like it should be renewable at the end of the cycle and available to further rounds of having as much protein of interest being targeted to the proteasome degradation pathway. Um,
But the flip side is if there is a covalent ligand that binds to the E3 ligase,
yes,
if you make a PROTAC out of that, now that's really interesting because now your PROTAC is already pre-bound to the E3
E3
ligase.
ligase. Yeah,
So
and
recruiting just the protein of interest makes it really attractive.
and are you looking at that, or is Dr. Zheng looking at that? Are there papers that are looking at that?
We are not, but I know that people are.
Yeah, a
Okay.
lot of people have published that. And actually, we have a paper online, manuscript online, use a natural product, covalent ligand for keep warm. So you actually see very effective degradation of target protein.
Can you tell me a little bit more about what is the covalent ligand and what does it do?
Yeah, we previously found we study a natural product called pepperlongomine for senescent cells. So we actually profiled the potential target for this natural product, and then we found actually several E3 ligase can be pulled down by this compound. So then we link this natural product to several different protein of interest ligand. So we see protein degradation very effectively.
Oh, that's really good. Okay, that clears up things a lot. Thank you, Dr. Zhang. And then the other questions: do speakers consider that a certain binding affinity is needed for the POI as a starting point for making PROTACs?
Maybe I can chime in. In my mind, I want to have a binding affinity below 1 micromolar. Recently, Professor Cruz, as a review, they mentioned you need probably below 0.5 micromolar.
Yeah, so I think for both yours and Dr. Natarajan's, it seemed to be that 1 micromolar is probably where most of the action is happening.
Yeah, there's a nice Cell review where they looked at this problem by studying all the kinase PROTACs. This came out, I think, late 2020. And I think they came to a completely different conclusion. They said that they were not able to see a correlation between binding affinity of the POI and the outcome of the PROTAC. So I think that's still an open question.
Yeah.
In my mind.
Yeah, completely agree. Yeah, I think it has to be case by case. But I think in general, when you consider, like a target, if was to start work on, you want to find a ligand, then you look at the ligand, maybe every ligand you find is like 20 micromolar. So probably the outcome may be pretty poor, right?
But this is mostly for the ligand that is being targeted for the E3 ligase. I think here he's talking about the affinity of the PROTAC for the protein of interest. But yes, I mean, like the and also I think, like, if you want to have something that is, like, dischargeable, as in you can just transfer it off to the proteasome degradation pathway, you don't want something that is very specific. So it's like striking that balance where it's able to bind the protein of interest but it's also able to release it, and finding that Goldilocks zone is a little bit of a challenge, and also the most interesting aspect of the research. Am I right in understanding that?
Yeah, to some extent, because we still, I don't think we have been able to uncouple the inhibition component from the degradation component. And the degradation component requires, as Dr. Zheng showed, you need a proximal lysine for it to be ubiquitinated. So until we are able to have a large enough data set where we can uncouple these things in a systematic manner, I don't think we can answer that question just yet. But you're right. You do want something that has an optimal K off so that the PROTAC comes off the protein of interest once the ternary complex is formed and the ubiquitination happens. So.
Okay. And another question, mature RBCs have no nucleus similar to platelet cells. Have you looked into the cytotoxic effects of DT2216 in RBCs?
Yeah,
And also, yes, yeah,
yeah,
I'll
we,
let
we
you
look
answer
at
that.
that, and we never see any toxicity to IBC. Um, yeah, we haven't. We have. That's a good suggestion. We haven't looked for anemia in animal models. You mean anemia animal model, or yeah, we don't see
Yes,
anemia, but we haven't used anemia models for the study.
well, yes. I mean, I think the idea is that it's since they don't have a nucleus, have we looked into the RBCs? Uh, But then, isn't it also true that the Bcl-XL is going to be targeting the mitochondria? So how does the cell having a nucleus? I mean, I'm asking, or as a layperson who is not able to understand why RBCs would be particularly more important than, let's say, WBCs. And I think I would probably ask as a more general: does it have off-target effects for? Uh,
Obviously, the PROTAC will degrade Bcl-XL in a majority of cells, right? If they
yeah.
have the complete UPS system. So, however, most normal cell does not under apoptosis stress, so they're not depend on those protein for survival.
Right. So
Yeah,
it's
it
only
actually
in this.
degrade
Yeah.
those protein, but it doesn't matter to them. I don't know how the functional change, but obviously they don't die.
And the idea of having this molecule and it doesn't cause any more stress. I mean, that's like, it's because that's a fine, you are introducing a foreign molecule. Does that it also induce some sort of toxicity, or it doesn't induce any toxicity?
Right. Yeah, that's why the phase 1 clinical trial will tell, what's the toxicity, what other toxicity we can observe. So those probably will be interesting to see.
Okay. Okay, I think we have finally got around to all the questions. Thank you, Dr. Zheng. Thank you, Dr. Natarajan, for a wonderful webinar. I enjoyed it and I learned a lot, and I'm sure that our audience member also did. Um, if you have any further questions, please consider reaching out to the speakers directly. Their emails are showing up on the screen. Um, as a reminder, the webinar will be archived on the DDN website, and you will receive an email notifying you when the webinar is available on demand. On behalf of DDN, I would like to thank our speakers, Dr. Natarajan and Dr. Zheng, as well as our sponsor BPS Bioscience. And of course, our audience, thanks to you for listening. Goodbye.
Thank you.
Thank you.